The overall goal of this procedure is to study metabolism using NADH imaging in a working heart preparation. This is accomplished by first isolating the rabbit heart. The second step of the procedure is to successfully cannulate all 4 chambers of the heart to achieve a working heart model. The final step is setting up imaging equipment and collecting NADH data at different pacing rates. Ultimately, results can show metabolic changes of the heart through imaging of fluoresced NADH.
The main advantage of this technique over existing methods like the Langendorf retroperfusion is that the heart is operating in its physiologic state with flow in the normal direction and with the heart providing its own pressure for coronary perfusion.
This method can help answer key questions in electrophysiology and the mechanisms of lethal arrhythmias. This is done by providing a physiological model to study the link between coronary flow, metabolism, and electrical activity.
Visual demonstration of this method is critical because vessel and heart chamber cannulation steps are difficult to learn. Improper technique can damage the heart and jeopardize the rest of the experiment.
Prior to running an experiment, rinse out all tubes and chambers of the working heart system with purified water, and then purge the water from the system by running the pumps. Add cellulose membrane filters in line with the Langendorf perfusion pump, left heart perfusion pump, and right heart perfusion pump. After adding perfusate to the system, perform a 2-point calibration for each pressure sensor.